A very good morning. Myself, Dr. Deshmukh, the head department of botany, Mahatma Fule Mahavidyalaya, Warud. Discuss the topic, the recombinant DNA technology, the new approach in biotechnology, or this recombinant technology and the genetic engineering. These are the recent discovery in the biotechnology. For that, recombinant DNA technology, as the name indicates, recombinant. That means the combination of the two different DNA molecule. One DNA they can cut and join with the other DNA. So in this way, we can form a combination of the two different DNA molecule. That we call the recombinant DNA technology. For that, when the recombinant DNA molecule is formed, he can speak, I am the hybrid DNA molecule. That means the combination of the two different DNA molecule, one that from plasmid and other that is genomic DNA, created by cutting and sealing, cut by the restriction enzyme, sealed by the DNA ligase. These are two enzymes having the different function, cutting and sealing. When introduced into the host cell, when these rDNA or recombinant DNA molecules are formed, introduced into the host cell, these are we call it as the factories of the cloning. A number of the recombinant DNA molecules are formed. Then multiplication, I multiply and code for the desired protein. So in this way, these recombinant DNA molecules are speak. I am the hybrid DNA molecule created by cutting and sealing. When introduced into the host cell, I multiply and code for the desired protein. Now this biotechnology, or for that history of the recombinant technology, actually there are having the two parts, BBC biotechnology and ABC biotechnology. The present day DNA technology has its roots in the experiment performed by Bohr and Cohen in 1973 in E. coli. There are two parts, BBC biotechnology, that means before Bohr and Cohen, that means 1973, before 1973, and ABC biotechnology after Bohr and Cohen, 1973. Now there are specially two tools, molecular tools for the recombinant DNA technology. For that recombinant technology, the first that is the restriction endonuclease. This is the restriction enzyme. That restriction enzyme having the function that we call a DNA cutting enzyme. This restriction enzyme, the source of the restriction enzyme, that is from the bacteria. All the restriction enzymes which are available in the market or in the world, they are made from a bacteria. Any type of the restriction enzyme, for example, EcoRI, Hindi second, PGV second, these are all the restriction enzyme. The source that is from the bacteria, first point. Second point, this restriction enzyme consists of the a 6 to 8 base pair sequence of nucleotide. For example, EcoRI, this is the restriction enzyme having the sequence ATT and TTA. So there are only 4 base pair is present. When suppose that restriction enzyme having the sequence not less than 4 and more than 8. जर समजा त्या रेस्ट्रिक्शन एन्झाईम मध्ये 4 पेक्षा कमी आणि 8 पेक्षा जास्त जर न्यूक्लियोटाइड जर असल्या समजून जायचं की तो रेस्ट्रिक्शन एन्झाईमचा सिक्वेन्स जो आहे तो नाहीये. देन this showing the a two-fold symmetry. So these are the restriction enzyme. May be from the sticky end, may be from the blunt end. A specific restriction enzyme they can form the sticky end, and a specific restriction enzyme they can form the blunt end. So on both the stand may be from the sticky end or the blunt end. That depends upon the action of the restriction enzyme. And second enzyme, that is the DNA ligase, having the function DNA sealing enzyme. They can join the phosphorus bond in between the two different DNA molecule. They can also, we call it as a diester linkage or the bond. So that is having the function of that DNA ligase. In IPC there are having the a class of the enzyme, and the last class, that is the ligase. So that having the function that is the DNA sealing enzyme. So these are the two molecular tools specially used for the recombinant DNA technology. Then the principle. The first point, introduction of the recombinant vector into the host cell. That means, suppose this is the plasmid. This is the plasmid. Now that plasmid, that we call it as the extra chromosomal double-stranded circular DNA having the ability of the self-replication. This is the plasmid. And most of the bacterial cell, these are the plasmid specially. Now suppose this is the plasmid. So you can cut this plasmid by using the action of the any type of the restriction enzyme. Just I told you that restriction enzyme having the specific sequence. ठरलेल्या restriction enzyme चा ठरलेला sequence असतो. आता when this you can use that restriction enzyme on the plasmid or the action of the restriction enzyme on the plasmid, they can cut that plasmid on both the side, maybe form the sticky end or maybe form the blunt end, or one side there are sticky end and other side there are blunt end, depends upon the action of the restriction enzyme. So second, that is the genomic DNA. Genomic DNA, that means a DNA which are that is interested to be inserted. ज्या DNA चा सिक्वेन्स आपल्याला माहिती आहे तो आपण त्याच्यामध्ये जो इंट्रोड्यूस करून राहिलेला आहे त्याला आपण जिनोमिक DNA जनरली असं म्हणतो. Plasmid वर action, when you can use the action on the plasmid of the restriction enzyme, same restriction enzyme also used on the genomic DNA. When suppose on the plasmid they can form the sticky end on both the side, then same restriction enzyme also forms the sticky end on both the side on the genomic DNA. Okay, then this genomic DNA then insert in the plasmid and then use the ligase enzyme. So there are a joining of the two different DNA molecule that can be takes place and forms the one combined, that we also call a recombined DNA molecule. That means there are combination of the two different DNA molecule, one that is from plasmid and other that is from the genomic having the specific sequence which are to be known. So here combination that will be takes place. Now once a recombinant DNA molecules are formed, introduced into the host cell for the multiplication, because the host cell, these are the factories of the cloning, and the plasmid, these are also called it as the vector or the cloning vehicle. Now when introduced into the host cell, then multiplication of that rDNA molecule, here multiplication of the rDNA molecules are takes place, and number of the desired product or the proteins they are formed. Now what is the restriction endonuclease? The bacterial enzymes, they can cut or split DNA at specific site. This is the function of the endonuclease restriction enzyme. Restriction endonuclease was first discovered in E. coli, restricting the replication of the bacteriophage by cutting the viral DNA. These are also called it as a restriction endonuclease or the genetic scissors or the molecular scissors that we call it as. For example, suppose this is a foreign DNA, both having the both the sides, these are the blunt end, and here restriction enzyme fragment, that means here a number of the fragments are formed. More than one restriction enzyme action takes place and forms the blunt end. Here below that, here that this is the action of that restriction enzyme form the sticky end, especially on both the side. And when introduced into the vector or in the plasmid, they can form the recombinant DNA. Then restriction enzyme, the nomenclature of the restriction enzyme, this is the important part. there are number of the restriction enzymes. For example, EcoRI. This is the name of the restriction enzyme. Here, the first letter of the enzyme indicates the genus name. For example, EcoRI. So that E stands for the Essentiae. Then CO, followed by the first two letter of the species, CO, that is from coli. Then comes the strain of the organism. For example, strain RY13R, and finally a Roman numerical indicating the order of the discovery. So this the nomenclature of the restriction enzyme. Just I told you all the restriction enzyme, the source that is from the bacteria. They shows the A2-fold symmetry, consists of the A4 to 8 base pair sequence of the nucleotide. Then these restriction enzyme having a cleavage side, that means the cutting side. Specific restriction enzyme is having the cleavage or the cutting side in the nomenclature. For example, EcoRI. This is the name of the restriction enzyme having the sequence AATT and the TTAA. So in that E, that is the genus name, first name, that is the genus. Then followed by the two names CO, that is the species. Then R, that is the strain, RY13, and first numerical nomenclature that we call numerical number, Roman numerical number. So this is the name of that restriction enzyme, EcoRI. Then for example, second enzyme, that is the Hindi third. So that Hindi third, Haemophilus H, influenza IN, strain RD D, and the third endonuclease, that's why we can written here, that is the Roman numerical third. So this is the nomenclature of that restriction enzyme. Now what are the cleavage side? Cleavage side, that is we also call it as the cutting side. It is the side where the DNA is cut by the restriction endonuclease. Just I told you that DNA that is cut by the restriction enzyme having the specific side, or specific restriction enzyme is having the specific sequence, and from that sequence only that restriction enzyme they can cut the DNA, forms the sticky or the blunt end. So it is the side where the DNA is cut by the restriction endonuclease. Restriction endonuclease can recognize DNA with a particular sequence of 4 to 8 nucleotide, consists of only 4 to 8 sequence of nucleotide. So this is the cleavage pattern of that restriction enzyme. If you observe minutely here, the scissors takes place here, and these are the restriction enzyme. For example, EcoRI, then BAM HI, then PST first, Hindi second. So these are the restriction enzyme, and these all the restriction enzyme having the specific sequence of nucleotide consists of the only a 4 to 8 base pair. So this is the sequence of that restriction enzyme. Now these restriction enzyme, now other one, that is the DNA ligase. So DNA ligase actively participate in the cellular DNA process, having the function joining of the two different DNA molecule. So that the DNA ligase joins or seals the DNA fragment by forming the, now this point is important, a phosphodiester bond. Now this phosphodiester bond, that means one that the DNA of the plasmid and other that is from the genomic. So that two different DNA molecules are joined from phosphate, so they can form the phosphodiester bond. So forming of the phosphodiester bond, that is the function of the DNA ligase, and that's when they can join the two different DNA molecule. Now the generation of the DNA fragments, now the principle of the recombinant DNA molecule, generation of the DNA fragment and the selection of the desired piece of the DNA, you can cut. So this is the plasmid, here this is the plasmid, That plasmid, extra chromosomal double stranded circular DNA having the ability of the self replication, and this is the DNA fragment. This is the genomic DNA, the green part, that is the genomic DNA. This is cut fragment from that eukaryotic chromosome. Now then second, insertion of the selected DNA into the cloning vector. So this is a plasmid and this is the genomic DNA. So that genomic DNA you can insert in the plasmid by cutting or by the action of the restriction enzyme. On both vector and the genomic DNA, only you can use the only one restriction enzyme. Those restriction enzyme which are used on the plasmid, same restriction enzyme is used on that genomic DNA. That's why they can form on both DNA molecule, sticky end or blunt end, on both the side, or one side sticky end and one side that is the blunt end. Now the host cell. What are the host cell? These host cells, these are called it as the factories of the cloning, because without the host cell the recombinant DNA molecule does not multiply. The living system of the cells in which these are the carriers of the recombinant DNA molecule or the vector can be propagated. For example, prokaryotes, the bacteria, and eukaryotes, the fungi, animals, and the plants. These are the host cells. Now, what is the vector? Now, this point we also called it as the cloning vehicle. Plasmid is called it as a vector. Just I told you that plasmid, that is the extra chromosomal double-stranded circular DNA having the ability of the self-replication, and in that plasmid only that is up to 5 kbp or 500 kbp the sequence of nucleotide is present, and in that plasmid when you can use the action of the restriction enzyme consists of only 4 to 8. So in that 5 kbp, 4 to 8 bp, that sequence of that restriction enzyme is found in the plasmid, and from that sequence that restriction enzyme cut the plasmid, and that's why the plasmid that we call it as a cloning vehicle or a vector. DNA molecule which can carry a foreign DNA fragment, they are self-replicating, no enzyme required for the replication in the plasmid. The plasmids, the vectors of these are the plasmid, bacteriophage, cosmids, and the artificial chromosome vector, especially the bacteria. Now the plasmid, these are the extra chromosomal double-stranded circular self-replicating DNA molecule. All the bacteria have the plasmid. This is the most example. The size of the plasmid vary from 1 to 500 kbp. This is the size of the plasmid, is circular DNA. Now the transformation. Once recombinant DNA molecules are formed, enter into the host cell for the multiplication, and in that host cell a circular DNA, in the host cell the host genome that is present, and from that host genome these rDNA molecules are multiplying. So these are the that is for the tools and technique of the recombinant DNA molecules. So thanks.